Hello everyone. In this video I'll be talking about amino acids and protein, and I'm following the organic chemistry textbook written by Janice Smith, and the chapter is 28th. So in this video lecture we'll be discussing about amino acids, synthesis of amino acids, separation of amino acids, then initial selective synthesis of amino acids. Then we will be discussing peptides, then peptide sequencing, then peptide synthesis, then automated peptide synthesis. And lastly, we will be discussing protein structure and then important proteins. So these are the topics that we will be covering in this video. First, we will discuss these amino acids. Let's see. So this is how you often see in the books the proteins are represented. So these are the ribbons. They are shown by this way, but they, as you know, the proteins are polypeptides. They are made up of several 100,000 amino acids depending upon the molecular weight of the protein. So myoglobin is a globular protein. So it's a type of a protein that is globular protein that contains 153 amino acids. So in myoglobin there are 153 amino acids joined together, as well as non-protein portion called heme unit. The heme group consists of a large nitrogen heterocycle complexed with iron cation. The Fe2 cation binds in oxygen. This one binds oxygen in blood and stores it in its tissues. Whales have a particularly high myoglobin concentration in the muscles. This is the picture of a whale. Okay, this is whale. It serves as an oxygen reservoir for the whale while it is submerged for long periods of time. In chapter 28, we discussed these properties of proteins and amino acids from which they are synthesized. Of the four major groups of biomolecules, number 1 is lipids, number 2 is carbohydrates, number 3 is nucleic acids, number 4 are proteins. So there are four big major classes of biomolecules: lipids, carbohydrates, proteins, nucleic acids. And proteins have the widest array of functions, such as keratin is a name of protein, collagen is a protein, for example, are part of large group of structural proteins. So these proteins, collagen and keratin, they provide structural functions that form long insoluble fibers giving strength and support to tissues. So these two proteins, collagen and keratin, are responsible for strength in tissues. Hair, horns, hooves, and fingernails are all made up of keratin. Collagen is found in bone, connective tissue, tendons, and cartilage. So collagen is a protein which is present in bone, connective tissue, tendons, and cartilage. Enzymes are proteins that catalyze and regulate all aspects of cellular functions. Membrane proteins transport small organic molecules and ions across cell membranes. Insulin, the hormone that regulates blood glucose levels, fibrinogen, and thrombin, which form blood clots, and hemoglobin, which transports oxygen from the lungs to tissues, are all protein. So you can see these are different variety of proteins that perform several different functions, ranging from keratin, collagen, enzymes, membrane proteins, insulin, fibrinogen, thrombin, hemoglobin. These are all proteins which perform wide variety of functions. So proteins are made up of amino acids. So amino acids are called amino acids because of this NH2 group, they are called amino. And because of this carboxylic acid, they are called acids. So acids because of this acid group, amino because of this NH2 group, alpha because this carbon is alpha. Okay, so this is alpha carbon, And then naturally occurring amino acids have an NH2 group to the alpha carbon of a carboxy group, and so they are called alpha amino acids. All proteins are polymers. This is a very important point. All proteins are polyamides formed by joining amino acids together. So a portion of protein molecule. So protein molecule contains this kind of portions. For example, this is one amino acids part joined together with another amino acid part. So 2, then this is the third part, and this is the fourth part, and this chain continues. That's why we put this wavy line, and this side also continues. That's why we put wavy line. And these are called portion of a protein molecule, and this is called protein structure. Now, general features of amino acids. The 20 amino acids that occur naturally in protein differ in identity of the R group to the alpha carbon. The R group is called side chain of the amino acid. The simplest amino acid called glycine, where R is equals to H. All other amino acids R is not equal to H. That's important point. So in all the other amino acids, R is not equal to H except glycine, and they have a stereogenic center, means they are chiral on the alpha carbon. As is true for monosaccharides, the prefix is D, this one, and L are used to designate a configuration at the stereocenter of amino acids. Common naturally occurring amino acids are called L amino acids. The enantiomers D amino acids are rarely found in nature. These general structures are shown in 20.81. This was the 28.1. According to RS designation, all L amino acids except cysteine have S configuration. So this is important. The cysteine amino acid has S configuration. All the other amino acids have R configuration. As you know that there can be R or S configuration. So only cysteine is S. Rest of the amino acids are R configuration. All amino acids have common names. The names can be represented by either a 1 letter or a 3 letter. Sometimes we write F, that means that is phenylalanine. Sometimes we write Y, that means tyrosine, that's a 1 letter code. And sometimes we write 3 letter abbreviation like G, L, Y. That is glycine. Okay, glycine. Listing of the 20 naturally occurring amino acids together with the abbreviations. Note the variability in the R group. A side chain can be a simple alkyl group, or it can have additional functional groups such as OH, SH, carboxylic acid, or NH2. Amino acids with an additional COOH group in the side chain are called acidic amino acids. Those with an additional basic N atom in their side chain are called basic amino acids. All other amino acids are neutral. So amino acids could be acidic, they could be basic, and amino acids can be neutral. So this is AA for amino acid. If it contains 2 or more than 2 COOH groups in the side chain, that is called acidic amino acid. If it contains more NH groups, then it is called basic amino acid. If it contains 1 acid group and 1 basic group, that is neutral amino acid. If you look at, this is the simplest amino acid, this is the glycine, which is achiral, it does not contain any stereocenters, and R is equals to H. There are 2 possible enantiomers when R is not equal to H, which is this case. So this is D amino acid and this is L amino acid. If you look at this one, this is NH2 group and this is acid group, and these are L amino acids and these are D amino acids. Only this isomer occurs in protein. So proteins contain L amino acids. These are the structures and the 1-letter and 3-letter code of the amino acids with their full name. For example, this is alanine. This is the structure of alanine. ALA is a 3-letter code, and this is a 1-letter code, which is A. Similarly, this is asparagine. This is the structure of asparagine. The code 3-letter for asparagine is ASN, and the 1-letter code is N. Cysteine, this is a structure of cysteine having the code CYS. This is the 1-letter code. Glutamine, that is the glutamine, GLN, that's the Q 1-letter code. Glycine, this is glycine, GLY, 1-letter code is G. Phenylalanine, that is the structure of phenylalanine, PHE, and this is our F. Proline, this is the structure of proline, PRO, and the 1-letter code is P. Serine, SER is a 3-letter code and S is the 1-letter code. Threonine, THR, and T is the 1-letter code. Tryptophan, TRP, that's the W is the 1-letter code. Isoleucine, ILE, I is the code. Leucine, LEU, L is the code. Methionine, MET, M is the code. Tyrosine, TYR, Y is the code. Valine, VAL, V is the code. Aspartic acid, ASP, D is the code. Glutamic acid, GLU, E is the code. Arginine, ARG, R is the code. Histidine, HIS, H is the code. Lysine, LYS, K is the code. Look closely at the structures of proline, isoleucine, and threonine. All amino acids are 1-degree amines except proline, which has its N atom in a 5-membered ring, making it a 2-degree amine. Isoleucine and threonine contain an additional stereogenic center at the beta carbon, so there are 4 possible stereoisomers, only one of which is naturally occurring. So this is proline, and this NH is secondary amine. This is isoleucine, which contains extra stereogenic center, is denoted with star, and this is L-threonine. It also contains extra stereogenic center. It's also denoted with star. So they also possess the extra stereogenic center. Humans can synthesize only 10 of the 20 amino acids. So the human body can synthesize 10 of the 20. The remaining 10 are called essential amino acids because they must be obtained from the diet. 10 amino acids which are present in our body, they are called non-essential, and 10 amino acids that we need to obtain from the diet, like eating food, carbohydrates and other things, and proteins, they are called essential amino acids. They are labeled with an asterisk in figure 28.2. So these ones are with star, they are essential amino acids. This is essential, this is essential, this one is essential. Valine is essential amino acid. Phenylalanine is an essential amino acid. Threonine is an essential amino acid. Tryptophan is an essential amino acid. Acid-base behavior of these amino acids. So amino acids, they occur in Zwitter ionic form. What is Zwitter ionic? In an ionic form which contains a negative charge and a positive charge on the same molecule. For example, this is carboxylate anion and this is ammonium cation. So this amino acid is neutral, NH2 is a base and carboxylic acid is a acid. So carboxylic acid donates the proton and becomes carboxylate anion, and NH2 picks the proton and becomes the ammonium cation. And the overall molecule is neutral because there is one positive and one negative charge. but that is called, the neutral form is called, the Zwitter ion is neutral. This neutral form of an amino acid does not really exist. This salt is the neutral form of an amino acid. This form exists at pH 6. Amino acids do not exist to any appreciable extent as an uncharged neutral compounds. They exist as salts, giving them the high melting points and making them water soluble. So due to the ionic form, they are soluble in water, and due to their ionic nature, they have the high melting points. Amino acids exist in different charge forms depending on the pH of the aqueous solution in which they are dissolved. For neutral amino acid, the overall charge is +1, 0 or -1. Only at pH 6 does the Zwitter ionic form exist. The carboxylic acid negative and ammonium group of amino acids are ionizable because they can lose a proton in aqueous solution. As a result, they have a different pKa values. The pKa of the acid group is typically 2, whereas that of ammonium ion is pKa is 9. Some amino acids such as aspartic acid and lysine have acidic or basic side chains. These additional ionizable groups complicate somewhat the acid-base behavior of these amino acids. pKa for these acidic and basic side chains as well. For example, this is how the charge of a neutral amino acid depends on the pH. So if the pH is 6, this form exists at pH 6. If the pH becomes 10, the overall charge will be negative. So this form is dominant. If you decrease the pH, make it acidic, then this form will be existing at acidic. So this form exists in acidic media, and this form exists in basic media, and this form exists at pH 6, which is almost neutral or slightly acidic. This is the table 28.1, which shows pKa values of ionizable functional groups of amino acids. So the alanine, the acid has pKa of 2.35, ammonia has 9.87, it does not contain any side chain, and the pi is 6.11. Same is for arginine, pKa is 2.01, for ammonium it's 9.04, side chain pKa is 12.48, and pi is 10.76. Same for aspargine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, tyrosine, valine. These are all amino acids and they contain different side chains. I am giving you a couple of minutes to look at this table and note down the pKa values before I move on to the next topic of this lecture. Table 28.1 also lists the isoelectric points, Pi. Isoelectric points for all the amino acids. Recall from section. Isoelectric point is the pH at which an amino acid exists primarily in its neutral form, and it can be calculated from the average of pKa values. So this Pi this Pi value indicates the pH at which all amino acids will be neutral. Okay, now let's have a look at the synthesis of amino acids. Amino acids can be prepared in a variety of ways in the laboratory. 3 methods are described, each of which is based on reactions learned in previous chapters. So SN2 reaction of alpha halo acids with ammonia. This is a halogen, this is acid, and this carbon is alpha carbon. If you react this compound with excess of ammonia, you will get amino acid. That's the one. Example of this reaction is this. So bromine is replaced with NH2, and this is how valine amino acid is synthesized. And you can prepare any amino acid by following this strategy, by reacting alpha halo acids with excess of ammonia. Although the alkylation of ammonia with simple alkyl halides does not generally afford high yields of primary amines, this reaction using alpha halo carboxylic acid does form the desired amino acids in good yields. In this case, the amino group in the product is both less basic and more sterically crowded than other 1-degree amines, so that a single alkylation occurs and the desired amino acid is obtained. Method number 2 is alkylation of a diethyl malonate. So this is diethyl malonate because this is ethyl group, this is ethyl, that's why we call diethyl, and this group is malonate group, that's why we call it diethyl malonate. So if you react this in 3 steps, we get to the amino acids. This reaction can be adapted to the synthesis of alpha amino acids by using a commercially available derivative of diethyl malonate as a starting material. This compound, diethyl acetamide malonate, has a nitrogen atom on the alpha carbon, which ultimately becomes the NH2 group on the alpha carbon of the amino acid. That's the one. So this is the diethyl acetamide malonate. So we have this group, and this one amide. So in 3 steps we get to this. That's how it reacts. So you react in the first step with the ethoxide base, which deprotonates this acidic proton, and the carbon gets the negative charge, and this carbon negative charge picks the R group from the alkyl halide via SN2 alkylation. So this is how R is attached, and in the next step we do hydrolysis and the heat to get rid of these ester groups. So ester is removed, this ester is removed, and this part is also deleted, and we get NH2, acid, and R. This step, last step, is called hydrolysis and decarboxylation. Step 1 is deprotonation, number 2 is alkylation, and step 3 is decarboxylation. So again, step 1 is deprotonation, step 2 is alkylation, and step 3 is hydrolysis and decarboxylation. Next is this phenylalanine, for example, can be synthesized as follows. So this is the structure which reacts with sodium ethoxide and alkyl part to generate this compound, which upon heating and hydrolysis gives you the phenylalanine amino acid. The last method of preparation which we are going to discuss in this video is Strecker synthesis. So this is a very famous reaction for the preparation of amino acids. The third method, the Strecker amino acid synthesis, converts an aldehyde into an amino acid by a 2-step sequence that adds 1 carbon atom to the aldehyde carbonyl. Treating an aldehyde with ammonium chloride and sodium cyanide first forms α-Amino nitrile, which can be hydrolyzed in aqueous acid to an amino acid. So this is aldehyde, this is ammonium chloride NaCN to form this α-Amino nitrile, which upon hydrolysis gives you the amino acid. The Strecker synthesis of alanine, for example, is as follows. This is aldehyde, which reacts with this group to give you the α-Amino nitrile, and in the last step, by the hydrolysis of cyanide, it gives you the alanine. So mechanism for the formation of α-Amino nitrile from an aldehyde. The first step in the Strecker synthesis consists of 2 parts. One is nucleophilic addition of ammonia to form an imine, followed by addition of cyanide to the carbon double bond nitrogen. Both parts are related to earlier mechanisms involving imines and cyanohydrins. So that's the first step. The lone pair of ammonia attacks on this carbonyl group, and the bond between carbon and oxygen shifts in the favor of oxygen. As a result, oxygen gets the negative charge, and there is a bond between carbon and nitrogen gets the positive charge because it has made 4 bonds. Nitrogen has made 4 bonds, that's why it is positive in charge, while oxygen has made just 1 bond, that's why it contains the negative charge. In the next step, proton transfer takes place from NH3 and it becomes NH2, and in the last step the OH group is removed and this bond reverts back and this OH group leaves as a water. So we get the imine formation, so like a shift base formation. And in the next step, nucleophilic attack of cyanide to form an alpha-amino nitrile. What happens? This nitrogen is deprotonated with HCl, and the cyanide acting as a nucleophile attacks on this carbon, and the double bond between carbon and nitrogen breaks in the favor of nitrogen. We get alpha-amino nitrile, that's the one. And in the last step, we do get the hydrolysis product of this cyanide to carboxylic acid. This is the reaction again to 3 methods of amino acids: SN2 reaction using an alpha-allocarboxylic acid, alkylation of diethyl malonate, and Strecker synthesis. So these are the 3 methods that we discussed for the preparation of amino acids. Number 1, that's the number 1. This is the alpha-halo carboxylic acid reacts with ammonia at large excess to give you this amino acid. Number 2 is alkylation of diethyl acetamide malonate to give you this amino acid, and lastly you react the aldehyde with sodium cyanide to generate nitriles, and upon hydrolysis it gives you the amino acid. So these are the 3 methods that can be employed to prepare amino acids in the laboratory. The details of the second step of the Strecker synthesis, the hydrolysis of nitrile to carboxylic acid, have already been discussed in this chapter. So we will discuss more points about this video, about these things, amino acids and protein, in the next video. And as we have covered in this video these 2 topics, the amino acids, what are amino acids, and their synthesis methods. So in this video we have covered these 2 points: what are amino acids and how they are prepared in the laboratory. Now in the next video I'll be talking about separation of amino acids, then we will discuss enantiomer selective synthesis of amino acids, and then we will make video, I will be making video about peptides, then peptide sequencing, peptide synthesis, automated peptides synthesis, protein structures and important proteins. So keep watching and I'll be making more videos about this, and I'm following the book Janice Smith, written organic chemistry textbook written by Janice Smith. Thanks for watching. Take care. Bye.